Alrighty. All right. So one of the things that happens during evolution is that something's changing, right? So the difference between me and a banana is, in its simplest terms, how would you describe that? How could you sort of say what's different at the most fundamental chemical level? Not, my phenotype or anything, but what is different really? Why am I different from a banana? Say again? Living and not living. Well, it is a different kind of life. My life as a banana I wouldn't be here. Or that would be a really weird day for you. What I'm getting at is that it's the DNA, right? Like if I had banana DNA, I'd be a banana. Now, I'm related to bananas, right? Because there are differences in my genome, in the sequence of letters in the DNA. So I don't make banana proteins. I mean, I do actually make some proteins that are the same as bananas, because bananas have some of the same things that they need to do that I need to do, which is if they need to make more cells, they've got to copy the DNA. What are they going to use to copy the DNA? They're going to use DNA polymerase. So I have DNA polymerase gene, they have a DNA polymerase gene, it's almost identical. So if you're changing your gene to evolve into different things, right, then that's achieved by making little changes in the DNA. Do you think that all parts of the genome evolve at the same rate? No. What kinds of things would be changing more slowly? Say again? Well, thinking about when the mutations actually happen, they're pretty random for the most part. But when they're selected for or against, right, then that's going to determine which things evolve more quickly, which proteins are changing faster. So what kinds of proteins would you not want to make big changes to? Or if you did, instead of becoming a banana, you'd just be dead. Say again? Potassium? Can you elaborate? Potassium is an ion, so what do you mean when you say that? What's that?
I said never mind.
Oh, but there is potassium in bananas, so I kind of see where you're going with that. That's true, and there's potassium in me. And I can kind of take that to a place, which is that we all have some similar conserved requirements, right? I need potassium to make my nerves work. Bananas need potassium for a different reason. But I have to copy my DNA. So I'm not going to go around messing with my DNA polymerase or, other machinery that everybody has too much, okay? But I could, for instance, toss out the lactase enzyme, and then I just have lactose intolerance, but I'm not dead, right? My point is that if you want to understand relationships between very distantly related organisms, what you're going to want to do is look at genes that don't change very much. Now, if you're thinking about categories of things that change rapidly and it doesn't matter too much what you do to them versus things that change very slowly, where do you think ribosomal RNA is going to fall in that spectrum? Slow, right? It's a machine that does this thing that all cells do. And they pretty much do the same thing that they've been doing since the beginning of time. So you can make small changes, and we know this because we know that ribosomes in eukaryotes are different from prokaryotes. So that you can make some changes and it's not going to destroy everything, but those changes will be slow, and there are going to be very few of them that you can make without having a big problem. So what he was doing was looking at ribosomal RNA, and as a result of that, he was looking at a bunch of different prokaryotes, and he saw that the sequence of ribosomal RNA between different prokaryotes differed more than he expected it to. And he was able to classify different things into different categories and said, oh, well, you know what? Based on the sequence of the ribosomal RNA, there's clearly a group of organisms, we're going to call them bacteria, and guess what? They also have some structural similarities, like they all have peptidoglycan, versus this other group that doesn't have peptidoglycan, still a prokaryote, and is, in fact, it turns out, much more similar to us than bacteria are. Okay, so here is a summary of some of the important features of these three kingdoms. So if we're looking at archaeans, then they are prokaryotes, meaning they have no nucleus. They don't have peptidoglycan. Instead, their cell wall has other things in it. And in comparison, bacteria, which are prokaryotes, do have peptidoglycan in their cell wall. And then eukaryotes are different again. If they have a cell wall, what's in it? It's not peptidoglycan. It's not like what the archaeans have. It's basically carbohydrates. It's basically glucose-based. It's either cellulose or chitin. Now, we've talked about this before, but this is just sort of to remind you of sort of the really big picture of how these three domains are related to each other. The idea is that the common ancestor of all three domains is about 3 billion years ago, when that early cell existed. And about 1.5 billion years ago, the bacteria and archaeans diverged from each other. The eukaryotes are thought to have been derived by an endosymbiotic process where a larger prokaryotic cell engulfed smaller ones and started this process of making organelles, basically. And there are some, as I've talked about before, some living examples of this kind of endosymbiosis that might mimic the way this would have happened early in evolutionary time. This you may be somewhat familiar with, or perhaps not, but these are the levels of classification. And so in other courses, you may have had to memorize them. You do have to, in this course, if you want the 1 point on the one question I might ask on the one test. So I might ask you if these classifications are put in the correct order. Since the whole chapter is about classification, it behooves you to actually know what the classes are and what order they appear in. So species, genus, family, order, class, phylum, kingdom. And I'm sure there's some fancy mnemonic that you could use to remember this. I don't have one to offer you. Usually I do, but in this case I don't. But I'm just going to warn you now that it's extremely likely I will ask you that type of question. All right, I'm going to focus today on species because the definition of species is different in eukaryotes versus in prokaryotes and in viruses. So, how do we classify eukaryotic species? Anybody know this? If you were talking about snakes or frogs or people, how can we tell if we're a different species? Can we tell just by looking? So this is the definition in eukaryotes. It is a group of closely related organisms that can interbreed. So basically it comes down to sex, right? If you can have viable, fertile progeny with another organism, then that is an individual that's in your same species. Here's an example of speciation within the group of bluebirds. These are all very similar looking but distinct species, and we know that they're distinct because you can't breed them and have viable offspring. If there were other hominids still living, then we would know that they were or were not part of Homo sapiens on that basis also. On the other hand, the prokaryotic world, we can't use that same definition. Why not? What's the the prokaryotic sort of equivalent of sex is this conjugation which may or may not ever happen. And in some bacteria don't do it at all. So, and most of them don't do it very often, even if they do. So it would be a really terrible way of defining species. I think the way that we define it in the textbook is kind of pretty terrible also, but here it is. A population of cells with similar characteristics. I don't like this definition very much, and I don't like it in part because in addition to being super vague, it's not really what's being used currently, okay? So actually, currently, the way we define species has to do with sequencing the DNA for the most part. But I'll get into those details in a second. Some terms that you need to know: culture, clone, and strain. So when we're working with stuff in this lab, we culture things, right? And that's just basically, it could be a mixed culture or it could be a pure culture. So what is an example of a mixed culture that we have cultured in this lab. When did we do a lab activity where we had a mixed culture or inoculum had several things in it? Yeah, when we did our environmental survey. And when we have used pure cultures, that's basically every other experiment we've ever done. A clone is a population of cells derived from a single cell. So what would an example of a clone be that you've seen? Yeah, colonies on a T-streak. Each colony would be a clone. And then this word strain is that some of the cells, even within a clone, can differ genetically from each other if during the process of cell division, the copying introduced a mistake, which does happen. And so it's actually, a strain is basically a variation within the same species. Now, a lot of times the way that we sort of functionally define species is that we're working with pure cultures, and we know for sure that they're related enough to each other to sort of be considered the same species, even if some of the cells within that culture are differed in some small ways. So when we classify prokaryotes, we use two different ways. We use phenotype, that is either their shape, the way that they do chemistry, or we can do it clinically using differences that we can detect using serological techniques or resistance to various kinds of drugs. The other way we do it, and this is much more common now in research, this is sort of done a lot in the clinic. This is done in research primarily, and it's genotyping. So we can genotype different prokaryotes in a variety of ways. I'm going to talk about 4 ways. So DNA fingerprinting, which used to be very common and now isn't as common. GC content, same thing, was common. DNA hybridization and sequencing, and not necessarily sequencing the entire genome, but sequencing, for instance, the ribosomal RNA.